So my name is Isogai. I'm a senior scientist at Allen Institute for Neurodynamics, and I'm a developer of Repix, one of the platform for chronic implant system, and especially we developed this so that this implant system is compatible with rodent social interactions. And I have a co-presenter today. Please introduce yourself, João.
Hi, and I'm João. I'm an assistant project scientist at UCLA, the Lab of Van Churchland. I mainly study decision making, and I use a device that we developed here called the INGI to record chronically from mice with multiple probes, and I hope to tell you a bit about it later.
Can you guys hear? Okay. Not very well. Maybe you want to try your microphone. Maybe.
Yeah. Let me try. Oh, if I speak a bit louder, is it better?
Yeah, that's better.
Yeah.
Okay. Anyway.
Okay. I'll try like that.
Okay, so today we are gonna go over the world of chronic Neuropixels recording, and I would like to start by asking you, how many of you are interested in recording chronically using Neuropixels? Raise your hands. Most of the people. So it's really amazing to see that the community really interested in this technology, at least that when I was starting at UCL, University College London, I think there were just very few people who were interested in doing this, and it was technically challenging problem. So really nice to see that we come to this point. So today we're just going to talk about a few things really quickly, and I'm going to hand over the presentation to João pretty quickly, and then so we visit a couple of different points. First is to discuss why we need a chronic paradigm. Maybe this is actually not particularly need convincing you about this, and then we move on to what are the requirements and what is really the state of the art. And we're going to go over the consideration of setting up chronic recording, and hopefully we have a few minutes for discussion. So first of all, why do we need to do chronic recording? And here we try to compare and contrast acute versus chronic recording. And obviously the major distinguishing factor between acute and chronic is that the chronic recording, the probe has to stay with the animal at all times. So that would entail a housing period when you are not recording the animal, recording from the animal. And acute recording tend to be used for head restrained behavior, and then chronic recording can be used for both head fixed state and also in freely behaving state. And because the animal has to be carrying the implant, so there's a weight limit, and I think there's a really big consideration for the animal mobility, and practically speaking, surgery takes longer. And then in terms of the performance of these implants, obviously the acute recording, the resampling of neurons in the same brain is straightforward, while chronic recording allows recording from the same brain for days, but it's not very easy to adjust recording sites. And then most importantly, we need to consider the activity degrades over time. And the gold standard for chronic implantation is directly cement the probes to the skull, but reuse tend to be very difficult, and probes could break for many reasons. And to enable the probe use, reuse, some of the researchers in the Neuropixels community over the years decided to develop retrievable chronic systems. So here I want to delineate some of the requirements of such systems. So I would put critical and optional, and really the high priority of the retrievable system would be the mechanical resilience. And this really depends on the applications that you're considering. But, to be simply put, animals will bump their head against hard surfaces, and this is really a really easy way to break the probes. And other applications, such as the applications that we're interested in, entails social interaction. So we need to consider mechanical stability during rough and tumble fights, for example. And it would be nice that these mechanical resilience can ensure long-term unit stability. And another thing that's really critical for our implant developers is to really ensure that the reproducibility of probe implantation and retrieval. And there are a couple optional criteria, but suffice to say that the really exciting development over the last few years is to be able to implant multiple probes into small rodents such as mice. And readjustability, like what you can do with tetros, will likely extend the utility of this system, and so that could be a really nice addition. And one area, such as tracking units over long periods of time, is an important feature, but it is currently unclear whether this can be done with retrievable implants. So to look at the, sorry. Okay, so to get at this question, to satisfy the essential and some of the optional requirements, all existing implants use a similar principle. And first, the components. So they typically consist of probe carrier and base, and they together basically are going to be inserted into the brain and with a fixture such as screws. Okay, so this is really challenging here. Yeah, so the whole unit is adhered to the skull with the cement. Okay, so to retrieve, basically simply detach this attachment, such as screws, and then basically pull this probe out. In some cases, you can actually readjust the probe by going up and down. So this is design, you might really know some familiar cases, which is lunar lander. So we call this a lunar lander design. And then we just want to quickly go over, there's a diverse collection of such implant system now existing in the community. So I don't really need to spend too much time here. So I think you can really read each of these papers listed here. But maybe I would just point out that all of them work really well, and they have been validated in different ways. So that would entail recording from different brain areas and also using different behaviors. And today we're really lucky to have João Couto share his state-of-the-art system, and that allows up to 6 probes to be implanted. So before I hand over to him, I'd like to talk about the general criteria for choosing an implant system. So really the major punchline here is that I recommend choosing an implant system by identifying which system has been used for experiments closest to your potential needs. So consider, for example, what is the target area? Do you need to implant more than one probe? How long do you intend to record? Do you need to track units over time? What is the weight of the animal to be implanted? How will you record the activities, head fixed freely behaving, solitary or freely behaving social? And most importantly, do you know anyone who's using one of the systems locally? So now I would just hand over the talk to João.
Thank you. Do you think that like this you'll be able to hear me, or do you want me to try to fiddle with the microphone a bit more? How does it feel in the room?
It's good. Okay.
Yeah, perfect. I can also hear you back. So feel free to just interrupt me, ask questions. It would be nice if we could make this a bit interactive. So I'm really excited to tell you about this device we developed, and I wanted to start by telling you that. Let's see if I can. Oh wait, that's on me. All right, I should have control now, right, you? Yeah. Okay, perfect. Sorry for the hiccup. So, I wanted to start telling you about a little bit about what the reasons that why we got into this business, basically. So originally, I come from being able to record from probes in an acute setting. And in that setting, you're really able to, you have a lot of flexibility. You can drive the probes wherever you want. You can target different areas simultaneously. You can have the same probe pass through different places that you care about. And in chronic implants, when we started, it wasn't really that clear that we could have this flexibility. And that stemmed from driving the probes individually, and also from being able to implant a probe and retrieve it in the same way that the probe got to you. So we didn't have to glue it. We wanted not to be able to glue anything, to have to glue anything to the probes, so that we could then reuse it maybe on an acute setting or all of these things. So to do that, together with Max Malin in Anne Churchland's lab, we developed the Indy. And the Indy has a feature that it lets you use the dovetail that comes with the probes and secures the probes just with the dovetail via this screw that is here on the back. And we try to optimize the surgery and all these procedures so that we could have a very tight interface between the cement and the probe, and also use a sealant that could encapsulate all the shanks, so that we could drive that. And I think Yo is probably now handing over some packages which have some of these fixtures, and I encourage you to like take them out of the package, try to break them, try to put them together. There are a couple of different parts. There's the main part that is this one here that you can see, and you can just like take it out, put it on a Stereotax holder and play with it. We actually developed, this is how it looks like. You probably have one on your hands, but we have a version for 1.0, and the one that you have is the one for 2.0 probes. And that's the one that we're going to talk more about now. There's a feature to this, which is that we actually are able to drive a probe in the holder using just our hands. The only thing that is kind of required for this is that you keep your hands together, or that you have like a solid surface where you could do this. And then for recurving the probes, you can also do it with your hands. Typically, this would require that you use the Stereotax, that you put the animal back on the Stereotax, and then you align the angles that you use to drive the probes so that you pull them out. Because we use the dovetail mechanism, then you can just slide the probe out using just our hand and reuse the probes in this way. So in this talk, I wanted to tell you a little bit of what opens up when we do this, and what we were able to achieve with using this device, but also tell you a little bit of the efforts that we did to quantify like whether recordings are stable, what the neuron yield is, and all these things. And then in the end of the presentation, I'll tell you a couple of tips for implanting multiple probes. And again, feel free to interrupt me. I'll just skip slides if we have to keep on track. So the first thing that we tried to do was to see how many probes can we implant in the same hemisphere of a mouse. And we're able to plan these trajectories very carefully and implant 6 probes in the same hemisphere. So these are targeting very specific structures. And in here, I'm showing you rasters from individual units. I wonder if you can see the mouse, Yo. Can you tell me if you can? Yeah, perfect. So then each one of these rows would be the activity from a specific trial for this unit. They're sorted by stimulus intensity, and this is during decision making. So you can see that some cells, for instance, this one in the superior colliculus, has a strong response to the stimulus that is dependent on the stimulus intensity. Other cells like to respond in other areas, like to respond during the period where the mouse reports its decision. And we hope that we can use this to kind of make strides into how decision making kind of like gets coordinated by the activity of different areas. The one important fact is that these probes with the fixture, they weigh 0.5 grams. So we're kind of in a position where we can put multiple probes together. This doesn't mean that we can put 6 probes in a freely moving mouse, because then the mouse has to carry not only the probes, but also the head stage. So when we implanted probes in freely moving mice, we implanted up to 4. And in here you can see the configuration of these probes. So there are 2 probes here, and then there are 2 probes over here. That's actually this bigger cap that you also have in this package that I sent you. And this cap is actually carrying the head stage. So then on the day of recording, we just have to connect the 2 wires, one on each side, and so we can record from these probes and from this animal. And what we did was we tried to put the mouse on this platform and have him explore the environment. The animal could just like walk around, and the black traces that you see here is what the animal explored. He went quite everywhere. And then I implanted the mouse and let him explore the same kind of platform or environment. You can see that at a certain point, he kind of moves this weighing boat that has water. He moves it to different places of the arena, and his behavior doesn't really seem hindered by having these 4 probes recording from his brain. And then we wondered, okay, so how much can we actually drive this through? How long can we record and track units? And I noticed that some of the animations might be a little bit off. But so what we did was we implanted a mouse, and then we started recording. Then we ended up having to go to SFN to present this work. So you see that there's a kind of a bump here where we didn't record. I go over to Portugal for New Year's, and then to New York to present the course, and we're still recording during this time. So we actually didn't really see a big decay in unique numbers. And over here you can see single units in red and multi-units on black. I like to highlight that this plot is in a linear scale. So usually you could plot this on a log scale if the decay would be very striking. But on a log scale, it would just look like really a flat line. So it's very encouraging that we can do this. It seems to be close to what we wanted to do. Now, we wondered 2 things from this point. We wondered first, on a single session, is there really any movement that we can see, or is this not present at all? So we started with a simple case, a case where we encourage a mouse to take a leap from a platform and to fall on the floor, and then we looked at whether we would see this on our recordings. And here I'm showing you the individual units, and if there would be any sort of artifact, we would see that this, for instance, this red unit here would have like a blip in the activity. It would probably go silent, and we don't see any of that. So the implant seems resistant to that. Then we also try to quantify this in a situation that is a bit more controlled, where we head fix the animal. And in this configuration, the animal is running on a treadmill. So there's plenty of room for the brain to move in relation to the skull, which would be the condition where we would evoke the largest movement artifacts. We then quantified movement using Dredge, which was developed by the Peninsula Lab to track motion. And over here, I'm plotting the trace of Dredge, so the motion estimate, and it's overlaid with kind of like a 2-scale picture of the probe. So you can see that the movement is actually less than the size of one site, and which kind of tells us we're in really a good ballpark for being able to record. Now from here, we're wondering, okay, so it seems to be stable, but now are we really losing anything by giving up our acute recordings? If the craniotomies are fresh versus if they're kind of old, do we lose signals along the way? So we decided to compare our data with data from the International Brain Laboratory, so data that was collected in acute settings. And because Max is actually recording during the IBL task, then we could actually compare trajectories that were similar while the mice were doing the same thing. So we grabbed the raw data, sorted it ourselves, and then tried to compare it. And this is what you see here. So we have the number of single units using exactly the same metric that we got in acute, that the IBL has in acute, and then the ones that we got with our chronic implants from the first week and also after 11 weeks. So we don't see a huge decay on this. We compared also the spike amplitudes. So these would be the spike amplitudes on these 2 acute recordings, and the spike amplitudes on 2 different animals that we used to compare here when they were just in the beginning after being planted and later. And then wondered, given that a lot of things can be changing during this. Just because we have a high number of units, it doesn't mean that they would be necessarily from the same location, right, in the brain, because there could be drift between the sessions. So we started by looking at individual sessions and then concatenating sessions that happen at different times and trying to estimate motion using Dredge, the same algorithm, but now across multiple sessions. When we do that, we see that the motion is not really that much across sessions, but it's hard to compare. We wouldn't know what to expect. So we used a data set collected by Anna Lebedev at UCL that is made open in the Neuropixels 2.0 paper, and we compared the motion that we get between sessions, between our recordings and their recordings. And we tried to subsample the sessions so that they're at equidistant days based on the days of Anna's recordings, so that it would be a fair comparison. We didn't see any difference. None of these are statistically different. We then also looked at the single unit yields between our implants and the cemented probes, and there seems to be a lot of variability across animals that is larger than the variability between using one implant or the other. So we're in a pretty good spot. I wanted to show you a summary plot of all the recordings that we did, but I'm going to quickly go over this. I'll just tell you that each dot in this session, in this plot, is actually a recording session that lasted for an hour. And I'm just showing you the number of single units that we detected using the same criteria across all the experiments. For instance, these 3 rows over here, there are 3 probes that were recorded in the same animal. These ones were recorded from the same animal, then retracted and implanted in another animal. We were wondering whether other labs could also reproduce this. So the lab of Wei Hong, right next to us, that does social behaviors, started using this. So Miming implanted 21 probes. He managed to recover many of those. It's quite funny, just relating to what Yo is just telling us, that for instance, these 2 probes here, they were not recovered, not because the shank was not intact. The shank was intact, but one of the mice ate the flex of the other mouse. So then there's some considerations to take when you're co-housing animals. And now if we can just start to look at what we need to do to design experiments and to plan experiments. So this is a bit of a highlight of the flexibility that we have on our recordings. So using the same fixture, we can record from many, many different areas in many different configurations. We do this by using a planning software. So before the experiment, we have a software that has the dimensions of the holder built in so that we can drive the probes in virtually to figure out whether they're going to collide or not, kind of similar to Pinpoint that you might be having a presentation on later, or that you had already. And because of these kind of like independent way that we drive the probes, we can also have probes that are, for instance, in this example, we drive the probes across hemispheres and skull clear one of the hemispheres so that we can do while we're driving the probes to record from deeper structures. And we can also have optogenetic fibers or fibers for optogenetics that we implant in the same craniotomy as where we have the probes. So at this stage, let's take stock of how we get the probes in. So I think we're going to be a little bit pressed on time, so I'll try to move forward quickly, and then you can just stop me at any point if you want. So the trick that we do is basically drill craniotomies as small as possible. We cover the dura after we insert the probe. We cover the dura with Duragel, and we cover the shanks with QuickSeal. Because we do that, then we can quickly just drop Metabond over the entire assembly, and we're good to go. We use Metabond because it dries really quickly, and it lets us just remove the probes quickly enough that if we need to implant another probe, it doesn't take us too long. This is how we do these things just on a standard stereotax, not the prettiest, but not the prettiest picture. But you can see the mouse over here. There are 2 probes that are being driven over here, and there are 2 stereotaxic arms. We're driving the probes quite a bit, and then in each one of these is covered in this way. We use a stereotax that has 2 manipulators because then we can drive the 2 probes down at the same time, which is one of the parts that takes the longest. So if we take a step back, we initially remove the skin and make sure that the skull is really, really clean and that there's no periosteum attached to it. I don't know if you can see it on the screen, but there are marks that are right over here. Is because I scored the skull using the scalpel. So you can see that there's kind of a grid on it We do this so that the cement really gets a good grip on it. You also see a screw that I drilled over the cerebellum, a part that I wasn't intending on recording from in this experiment, and we use that for grounding. I implant a ground screw on that craniotomy and mark the craniotomy that I'm going to want to perform in the future for driving the probe through. You might notice that the craniotomy is elliptical, and that's to minimize the open area. The basic thought behind this is that if the craniotomy is small, the brain will not have so much room to move around, and it will not cause so much damage as might be important for when you're having multiple probes in. We then cover the entire thing with Metabond. We do that so that, for instance, if we put saline over the craniotomy, the saline is not in contact with hair that might be around and might be contaminated. So we try to maintain aseptic conditions in this way. We then go back to the craniotomy and drill it by thinning very, very much so. I don't know if you can see it on the picture, but there's a blood vessel right here. So that's when we know that when you start seeing the blood vessels, that's when we know that we're really close to the point where we don't want to drill anymore. We just want to put moist on the craniotomy, so saline or artificial cerebrospinal fluid. And then we can use a pair of forceps to open the craniotomy. We try very carefully not to touch the brain, and we try to leave the dura intact. We then sharpen probes. So I think you have a demo session for sharpening probes. I would encourage you to always sharpen probes when you do chronic recordings, not because you won't be able to pass through the dura of mice necessarily, but because if you pass through the dura a bit more aggressively, it might be that you're in a situation where there's a dip of the brain, and because you're applying Quickseal, like you'll see later, you won't be able to relax. The tissue won't be able to relax, and you'll be in a condition where you're not really in a healthy state. So now, one thing that is also important is that we drive the probes very, very slowly. We drive them at 1 to 5 micrometers per second. And we do so, and that has been correlated with the amount of units that we get. So I would really go slowly with this. And then after the probes are in place, we can use Quickseal to cover the whole shank. We do this by applying it with a dental applicator. Actually, this is something you can get from Amazon for really cheap. We backfill a syringe with Quickseal and then drive it around the shank as much as needed so that the entire shank is covered. At this stage, we can just apply Metabond. The viscosity of Metabond is really important, so I would recommend that you use a cold dish and you mix Metabond with this kind of mixture on the dish and apply it then. We've tried different ones, and some work very well, some work very badly. And then the last thing I wanted to touch before we do this is how it looks like when you're doing a surgery which has more than 2 probes. So when you have more than 2 probes, you might have to cover craniotomies, and you're implanting them 2 at a time. You might have to cover the craniotomies at a certain point. We usually cover the craniotomies using just Kim wipes that were sterilized, and that allows us to dry the craniotomy, so we can implant release the Quickseal to cover the shanks, put the Metabond, release the Stereotax manipulators, and then uncover 1 of the craniotomies or 2 of the craniotomies and implant them further again from there. A last thing, just to tell you how the recovery looks like. So this would be an example of a mouse that has been implanted with 6 probes. This is the day after the recording. You can see that the mouse can walk around quite nicely, given that he had a surgery just the day before that lasted 6 hours. And we do give drugs to the mouse to both minimize swelling, reduce pain and inflammation, and as sort of an antibiotic to prevent inflammation and or infection. Yeah, with this, I wanted to thank you all. I wanted to thank the organizers and also Yo, and I wanted to highlight my collaborators for this design, and also some people that helped us in the early beginning of the design, of when we were trying to figure out like how we're going to do this, and we asked help to everybody. So the bioarchive, our preprint is over here, and our designs and instructions are over here. If you have any questions, just reach out. And yeah, maybe we could go into a round of questions.
All right, thanks.
Thank you.
have the products or the holder like with camera. Like free moving recording near the camera, either for eye tracking or like word camera.
So let me just see if I understood the question. So you're wondering, like, for instance, in this case, whether the holder is impacting the in this case, whether the holder is impacting animal tracking or behavioral tracking.
Because working on like video processing, so we need track our eye movement, and also we would like to know where is our visual stimulus. So that's
Yes.
why we need two camera, at least one camera.
Yeah,
and then we don't want to really increase the weight because we are also use the nature model.
Yeah, no, that sounds great. So if you don't want to increase the weight, there was one solution that I thought was quite neat. The couple labs actually did this by now, but there are also head-mounted cameras that one could use. For instance, the lab of Chris Neal developed one that is really nice. It's documented in a paper where the first author is Parker, and in this case, you could actually use a camera to look at the eye and see where the eye is, and measure the pupil, but also another camera that would be pointing at the world so that you could see where the mouse is looking at. There's a clever trick to calibrate these two. This would limit the weight, obviously. So if you want to do this from outside, I think it would be quite challenging to still be able to monitor eye position. But you could certainly find a configuration where you can still see sort of the face of the animal if the stimulus is always in the same place, and track its position on an arena from above.
But still, the camera just increased a lot of weight. I went to Chris Neal's lab to see, and like basically female it's really hard to carry everything there. So that's why. But I heard someone mention to me, like it seems the Neuropixel probe has the camera inside. I don't know. They didn't mention. Yeah. That's why I'm just wondering if there's something possibility, like really decrease the weight.
No, I mean I'm not familiar with having a camera built into the thing. You could think maybe there's a way of miniaturizing the head stage a bit further to include sort of whatever's needed to drive a camera as well. But yeah, I'm not familiar with that.
Okay, thank you.
Thank you.
All right, thanks, Joe.
Thank you. Thanks, everyone. Bye-bye.